under microscope. And the inference part, we actually sample the cell shapes from the database, feed it to a mesh generator which creates a synthetic map. And then the synthetic map is further fed to the generator where we get the output, our synthetic image. For the conditional, again, we utilize piece-to-piece framework, which is the work presented at the last year's CVPR. The idea here is that you have a semantic map as an input. In this case, the segmentation map of the city in a sense. And then they utilize this method to translate the semantic map to the photorealistic images of the the city. In our case, it's in the same concept, but instead of their semantic map, we utilize our semantic map. And also, the cool feature of this network is that we can, there is a feature in photo part which can be used to play with different styles. So we can differentiate these different styles of the images. Once we train the first row is the ground truth image that we use for annotation. The annotation image. The second row is the ground truth image, and the third one is generated images. So once we train the piece-to-piece framework on the semantic image and the real image correspondence, we could generate the images in the third row. The cool thing here is that the network learned what the red blood is, so we can see that it can actually generate different circles in a sense. And the cool thing here is that the generated images are actually very different from the ground truth. So we can use our some hints that these images might be really meaningful for extending the data set. Also, we can play with the different styles, so we can generate different style, a different amount of noise in the synthetic images. The other thing is that we want to generate as many as possible semantic maps. So the most easiest and the most obvious way to generate the semantic maps would be just to place randomly cell shapes on the image. But actually, due to the concept called cell adhesion, the cells tend to stick to each other, forming a cluster, so like cluttered objects. So we come up with a different strategy where while we sample the shapes from our database, we actually create a probability density function which corresponds to the location where the cells should be placed. So it's an iterative process, and while we place the cell, we also update the probability map of the next location where the cell should be placed. And then it goes on like this until you pretty much place the number of cells you want to place on the image. So as a comparison, we compare the 500 cells placed randomly, which you can observe on the left bottom there. And also, right there, you can see the same amount of cells placed using our method. And on the right, you can see that, on the right, the left row is actually all synthetic semantic maps. And then on the right, you get all synthetic output. So you have synthetic input in, synthetic output out. So we utilize these images further to train on several tests. So we train it on segmentation. We use it in the segmentation test and detection test. And also there is another one, detection for segmentation, that is based on the segmentation. The key observation here that we observed is that actually for segmentation, we observed really marginal results, which could be actually in the range of statistical error. But for the detection, we observed 1-2% percentage points increase in the AP, the average precision metric. And so the key conclusion that we obtained from this work is that synthetic data overall has a promising potential, and the method that we developed is actually suitable for offline augmentation because in our case right now the unoptimized code takes about 1 minute to produce segmentation mask and as well as the synthetic image. And generally speaking, depending on your purpose, the utilization of the GANs and all this synthetic image process generation might not justify the fact might not justify the performance you want. So, for example, if you're trying to distinguish between like cats and dogs, then that 1 or 2% is not really significant. But if you're just trying to detect some diseases like self-infected malaria, for example, then that 1 or 2% is actually about saving lives. So, yeah, depending on the purpose, the method could be helpful or not, I would say. Okay, thank you for listening.
So, because of time, we're just going to move on to the next speaker. All of the presentations here are also going to be at the posters. So, if you want to ask a deep discussion, please take a question.
그냥 간 쓴다는 얘기 말고는 아무것도 지금 얘기 없잖아.
네, DC간.
어?
DC간.
So our next speaker is Tapegado Rai.